Hello everyone, and welcome to today's webinar. My name is Sherlyn Chen from Boehringer Ingelheim, and I will be your moderator for today's webinar that is organized by the GastroPlus user group. This is our second webinar since the user group established last year. Today's presentation is titled Quality by Design and Biopharmaceutics, Application of Absorption Modeling. Our presenter is Dr. Philippos Kasesoglu from Merck Research Laboratories. Philippos is a senior principal scientist in the Biopharmaceutics group, Pharmaceutical Sciences, and the Clinical Supply at Merck, where he's leading the modeling and simulation quantitative biopharmaceutics and oral biopharmaceutics efforts. His research work and responsibilities include the in vitro, in silico, and in vivo biopharmaceutical evaluation of oral formulations across development phases, focusing on the development and application of absorption modeling and its integration with preclinical data and other modeling simulation tools in a cross-functional setting to facilitate formulation decisions. Also participating as a panelist today is Dr. Vera Lukchova from Simulations Plus. Vera is a team leader for simulation technologies and also serves as a GastroPlus product manager. Following the presentation, there will be an opportunity to ask questions. You may either send your question using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. If you are using a telephone to listen to the call, please be sure to enter the unique audio pin displayed when you join the call. This enables us to unmute your line so that we can hear you. This webinar will be recorded and will be available to the users group members in the future. So without further ado, I'm glad to present Dr. Philippos Kasesoglu to start his presentation now. Philippos.
All right. Thank you, Sherlyn, for the introduction. Hopefully everyone can hear me okay. I'm really excited to present today the second GastroPlus user group webinar. I really hope that this presentation will serve as a starting point for a wider dialogue within our user group on the application of absorption modeling in quality by design. The underlying theme of the webinar today is understanding how the formulation properties relate to the clinical performance, often judged by the pharmacokinetic profile. The critical step to this, as we all know, is understanding how the formulation behaves in the gastrointestinal tract. To many outside the formulation or the biopharmaceutics area, the GI tract is treated like a black box. At the same time, as we'll be discussing modeling today, I also want to clarify, we don't want to replace this knowledge black box with another black box in the form of a computer program. So as we'll be talking throughout this webinar about understanding the formulation performance, I think it's equally important to keep in mind the need for understanding of the models we'll be using. A few years back, I had the opportunity to participate in a workshop around quality by design and biopharmaceutics. The outcome of this workshop is summarized in this picture shown on the screen. These circles, these 5 circles, show how the patient benefit, which is at the center, which is the end point of quality by design, links to a wide array of activities or tools that we're commonly using in the pharmaceutical development process. The webinar today will focus on the aspects of this red purple circle, the in silico tools. But as you will see throughout the webinar, the modeling activities do touch upon many other areas and are not disconnected from the rest of the activities. I will also encourage all the participants, if they're interested to learn more about this concept, to follow these publications that the citation is given at the bottom of the slide. However, I'm going to highlight two important aspects as related to quality by design and biopharmaceutics that we'll be discussing today. First of all, to many people, quality by design translates to a late-stage development activity. However, especially when we're talking about quality by design and biopharmaceutics, this should not be the case. We should be building the knowledge in a continuous manner from early development to late development. Second, it's again very critical to keep in mind that achieving the quality by design goals, it's an interdisciplinary effort and requires collaboration and integration of knowledge and data across many different assays and across many different groups within each company. So even though we'll be discussing modeling today, again, this job should not be seen as solely the job of the modeling group, wherever that group may reside in their respective organizations. With this general introduction to quality by design and biopharmaceutics, I'm going to shift gears and talk a little bit about oral absorption in this introduction. There is no doubt oral absorption of compounds is sufficiently complicated. Different aspects of oral absorption may be of different interest to the different groups within each pharmaceutical company. For example, a drug metabolism person may have a separate view on the oral absorption compared to a formulation or a biopharmaceutic scientist. However, as far as formulation development goes, that we will be discussing today, the primary concern is, at the end, to develop a robust formulation. This involves answering questions around what API phase to use, what will be the properties of this API, what excipients we should choose, and what manufacturing process, etc. Many of these are typically probed by dissolution assays. Unfortunately, oral absorption is a little bit more complicated than the glass dissolution beaker, and there's a lot of interactions between the formulation parameters and physiological parameters. This interaction can really dictate the final behavior of the formulation in vivo. So I'm sure this question is familiar to many people on the call. It's a situation often encountered. We always ask the question, I got these three profiles on the left, what do they translate in terms of a PK response? Are they going to be all similar, all different, or something in between? It's really practically impossible to test all the possible formulation variations clinically. I mean, if it's three, you can test them clinically, but typically we have more than three variations when we look at formulation development. So we do need alternate ways to understand the formulation performance. This understanding of product performance really comes through knowledge integration. We fortunately have a host of tools at our disposal to study formulations. These range from in vitro assays, symbol, solubility, and other physicochemical property measurements, dissolution studies, etc. In silico tools, and then eventually in vivo studies, either preclinically or clinically. What is important is to keep in mind that the information direction should not be one-directional. As more data are gathered, and as you move towards in vivo data, this data should be used to refine the models and the assay selection in a learn and confirm manner. Oral absorption PBPK modeling is one of these available in silico tools. I'm sure everyone on this webinar is very familiar with what absorption modeling is. Just simplistically, as a purpose of an introduction, oral absorption for the purpose of this discussion will refer to a mathematical combination for a given compound of the dissolution process, the absorption process, and the systemic disposition, shown on the left-hand side, combined with gastrointestinal physiological parameters to predict either the oral time course profile of absorption or the entire plasma concentration time profile. The concept that will be discussed today can be applied with whatever software is your favorite software within each organization. Most of the models that will be discussed have been developed in GastroPlus, but again, based on each one's experience, you can select your favorite software. When discussing linking the product profile, dissolution, and the performance, the first thing that comes to everyone's mind is an IV-IVC, an in vitro in vivo correlation. So why are we discussing absorption modeling today, and we're not discussing an IV-IVC? To avoid any confusion, I do believe that the IV-IVC, based on traditional approaches, and at least as far as regulatory applications go, will remain the gold standard, at least for the foreseeable future. However, unfortunately, pursuing IV-IVCs is not easy, especially earlier in development. To start with, you do require clinical data on different formulations. Especially for immediate release products, it's quite difficult to match the in vivo absorption and the dissolution timeframes, or to develop dissolution methods that are sufficiently discriminating given the very low solubility, and at the same time, they're also biorelevant. Therefore, we need some alternate ways to establish these IV-IVCs. And the absorption modeling today I will be presenting is positioned as an alternative to this traditional IV-IVC approach. In fact, some type of IV-IVC is an underlying assumption in all these PBPK models, as you are always extrapolating data from in vitro measurements to in vivo. With this introduction, to bring everyone on the same page around this webinar, I'm going to discuss a few case studies around areas where absorption modeling can be linked to quality by design discussions and questions. Again, although the majority of these models that will be discussed were developed in GastroPlus, you can develop similar models with your preferred software. The first case study we will be discussing is around understanding the impact of API particle size on absorption. This is a quite common question raised during formulation development, as well as at filing stages for specification setting. In this case, the model I will describe was taking place concurrent to the formulation development activities. The compound studied is a BCS class 2 compound with relatively low solubility in the intestine, although you can argue it's still decent solubility, and high permeability. For our first in-human studies, we had used jet milled API, so the particle size was a couple of microns, and we got good exposures. As we were moving towards later stage development, we wanted to understand what would be the impact of a larger API on pharmacokinetics, and whether the milling step, which obviously adds additional cost, is really required. At the time we got involved from the Biopharm group in the project, there were data available for the API dissolution of jet milled and unmilled material shown on the right. The first step was to assess whether we could predict the behavior of the API in silico. On the left-hand side, I'm showing a couple of simulations of in vitro dissolution using the GastroPlus software, where we simulated the micronized, the unmilled material, and a bunch of test lots where we had generated as potential API lots to move to later development. The model, the API diffusion-based model, gives a pretty good agreement between the simulated data on the left and the observed data on the right for the micronized and the unmailed API. I will add this is generally expected when one is looking at the dissolution of an API powder. You can see the rest of the simulations on the left, the rest of the lots span a pretty wide range of dissolution profiles. And some of them look pretty close to the unmailed material. So the question is, will this impact PK if this is true? We had clinical data for the compound at the time we got involved with this question. So building the model is relatively straightforward, using API particle size, solubility, permeability, and the observed PK parameters. Generally, the model does a pretty good job capturing the profile as shown on the graph, although I will admit that we are lagging a little bit on the absorption phase. Regardless, when we plugged in the rest of the test API lots, the particle sizes, distributions in there, you can see that the predictions are not drastically different from the observed data. There are small deviations, about 10 to 15% at worst case, on the PK. This will still be a viable option based on our understanding of the PK and PD of the compound. However, up to that point, we're solely working based on API particle sizes, ignoring the formulation factors. And in many publications, if you look through the literature, the API particle-based dissolution model is very commonly used and assumed that it's operating in vivo. However, one needs to be cautious about this. In our experience, the dissolution of the formulated API almost never is identical to the dissolution of API powder. The two graphs show the dissolution of the API powder on the left and the dissolution of the formulation for the same API on the right. Clearly, after formulation, the macronized API dissolves slower. Thus, to simulate this behavior of the formulation in vivo, we need to adjust for a formulation factor. There are multiple ways to approach this, including using a model such as the Z-factor model in gastroplasm. In our case, we chose to use a correction factor on the diffusion coefficient that allowed us to describe the new formulation dissolution profile. The obvious question at that stage was whether the dissolution correction factor is specific to the macronized API lot or it's universal across API sizes. Before we asked our formulation colleagues to go and generate formulations for every possible particle size, we're going to test whether these slow down, if true across all API lots, will be biorelevant, meaning it will have a PK impact. So we applied a universal correction factor to all the dissolution profiles. As you can see in the graph, the blue lines are the macronized data I showed before, and it shows you the shift between API and formulation. And the rest of the dashed lines are the predicted slow dissolution profiles. Once we plug this in our PK model, in our absorption model, we see that as far as the macronized lot, whether we use the API-based dissolution model or the formulation dissolution model, there is no practical impact on the prediction. The curves are almost superimposable. That's the blue line on the screen. However, for the rest of the lots, there is clearly an impact in terms of slowing down absorption and eventually even losing exposure if the dissolution does become very slow. So we had sufficient evidence to suggest to our formulation group to go back and make these formulations and test them at least in the dissolution first. The dissolution for the same lots, all of them is shown on the left-hand side. all the dissolutions at the end are contained within what we have previously simulated areas of being within bioequivalence or at least acceptable PK behavior. I will add that some of these profiles, for example, if you compare the yellow and the blue curve, will still probably fail traditional similarity criteria like F2. So from a dissolution standpoint, this will still be considered different. However, from a PK standpoint, our model suggests that this will be adequate for clinical performance. What we saw is obvious from the slide that the smaller particle sizes actually dissolve slower compared to the API, while the larger particle sizes actually speed up in the dissolution from the formulation compared to the API powder. Therefore, based on our model and our understanding, and along with the dissolution data, we felt confident that we could move forward with more relaxed API requirements. Unfortunately, the program was not advanced much more past this stage to really apply these findings in an actual specification setting. However, I'm going to recap case study 1 with some lessons learned. Although one could argue that these were quite basic models, and these are not extremely difficult models to run, I will say that the model was a very useful tool in terms of communicating our view of translating the dissolution data to a clinical impact in other areas outside biopharmaceutics, where they might be less familiar with these tools, and also to drive resource utilization. Again, while this work was done earlier in development, the learnings and similar principles can be easily carried out to late-stage activities and toward the specification setting. I did highlight that the API particle size distribution-based dissolution models do not always match the formulated product dissolution. However, I do think they provide a reasonable starting point, especially in early development. Finally, as shown in this case for the micronized API lot, whether we used an API-based model or a formulation dissolution model, the final outcome in terms of the PK profile was identical. Obviously, these are two different models with different assumptions. Therefore, based solely on the outcome of the model, it's difficult to declare whether we have a unique, correct model for the behavior of the compound. I will circle back to this point around model validation at the end of the webinar. The second case study I'm going to discuss today deals with understanding the relevance of dissolution conditions and how one can use absorption modeling to potentially develop and define a biorelevant dissolution method. The compound studied is a toricoxib. It's a BCS class 2 weak base. The physicochemical properties are shown on the slide. It's freely soluble in the stomach. However, it has relatively low solubility in the intestinal pH range. The reason for this investigation was the results of multimedia dissolution experiments that are commonly conducted when one is faced with manufacturing changes or to answer potential bioequivalence questions. The graphs on the right show the dissolution profiles we got for different batches manufactured at two different sites across different potencies of the product. It's evident that the pH 1.2, all the profiles are basically superimposable with no meaningful differences. However, at the pH 4.5 and pH 6.8 dissolution data, there is a clear dissolution difference between the supplies in the new site compared to the older site, and these fail F2 equivalence criteria. To develop a PK model, the first step was to describe the absorption of a toricoxib. We have internal IV data for the compound, so specifying the PK model is pretty straightforward. And again, using the standard inputs such as permeability, solubility, and the observed dissolution rates, we first validated the behavior of the model against clinical data. This was done across doses. I'm showing only the highest dose here, which will be the most sensitive as far as absorption goes. Validate against the outcome of 2 different studies with 4 different lots. The model describes the outcome, the clinical behavior pretty well. On the right, I have plotted the model predictions for the in vivo absorption and dissolution time course. What the model suggests that is happening is that the drug largely goes into the solution in the stomach, and then due to absorption, it doesn't precipitate much in the intestine until eventually the dose is absorbed. So if the stomach dissolution is driving exposures, we wouldn't have to worry about our signal in the dissolution data at the pH 4.5 and 6.8. This will be irrelevant to the clinical performance. However, as a worst-case scenario, we're going to actually perform projections, assuming this dissolution data drive the product performance, and see what would be the outcome of a potential bioequivalence study. The intent then was, do we take the risk of running such a study or not? The projections are shown on the top table. We ran a virtual simulation, a virtual trial on these different lots. And you can see that if we believe the difference is seen at pH 4.5 and 6.8, we're projecting up to 14% differences, especially on Cmax for the 2 profiles. And with the new site dissolution being higher, this will result in a higher Cmax, which could be a safety concern, theoretically. The differences are not large. One can design a bioequivalence study to pass a 1.14 and 1.11 GMR, but still, we wanted to see what would be the outcome here. Because of the regulations when we were dealing with this product change, we actually did have to run a clinical bioequivalence study for these 2 lots. The data are shown on the bottom table. It's clearly from the clinical BE data that the pH 1.2 dissolution is more clinically relevant compared with the pH 4.5 and pH 6.8 dissolution data. You can see that the geometric means for either AUC or Cmax are very close to 1.0 and with pretty tight confidence intervals. So clearly the dissolution difference is projected and the PK difference projected by the dissolution at the intestinal pH do not appear to be clinically impactful. So summarizing the lessons learned, for toricoxib, the dissolution differences seen at the higher pH were not reflecting the clinical bioequivalence study. The same can happen for other weak bases that dissolve quickly. Therefore, the standard F2 similarity criteria at the intestinal pH values may not be too meaningful in terms of projecting clinical performance. In this case, we were actually conducting the simulations to project POS of a bioequivalence study. Perhaps in the future, such models can be also used once there is sufficient comfort level with using these tools to argue for biowaivers for BCS class 2 and weak-based compounds, and potentially other BCS classifications as already have been published in the literature. For the third case study, I'm going to shift gears a little bit. So I'll discuss simulations, not on an average basis, but rather in the context of population variability. Still dealing with a CMC question, however. The CMC question at hand is one commonly encountered. What is the impact of a conversion of the salt to the free base in the formulation on bioperformance? The compound we'll be discussing, it's a weak base. It's again freely soluble in the stomach. It's extremely insoluble in even simulating intestinal media. Its dose is the hydrochloride salt, and the hydrochloride salt is known to have high bioavailability, almost complete absorption, and it behaves like that regardless of the stomach pH. The initial simulations I will show here were performed with SimShip using the population simulator. And I will get back to transferring some of these models to GastroPlus in subsequent slides. None of the currently available software, unless you write your own software, allow for the presence of two API forms simultaneously during the absorption phase. And I will get back to this point later in the discussion. So this initial simulation was run in a stepwise fashion. First, the part of the hydrochloride salt was simulated as a non-precipitating solution. Then a subsequent simulation was run for the free base portion based on the pH solubility profile. To get an initial estimate of the effect of these two profiles were just added together, assuming superposition, realizing this is an approximation. It does work relatively well in this case due to the very low intestinal solubility. So I'm showing here the 20% free base content simulation as a discussion point. The goal of the simulation is really to understand the impact of stomach pH on exposure at each given free base content, percent free base content. The top graph shows the outcome of all the simulations as far as fraction absorbed goes, or you can plot similar things for AUC or any other PK parameter you may be interested in as a function of stomach pH in the simulations. The as-per value is pretty low. So one may conclude that pH is really not of a significant impact here. However, this view is somewhat misleading. To get a better understanding of the data on the top graph were replotted on the bottom graph. So these are exactly similar, identical simulations, where the y-axis now is normalized to what the simulation projects in the same subjects for 100% hydrochloride salt formulation. It's clear that now there is a much stronger correlation. The R-square significantly improves. This way, this replotting of the data removes other factors that result in intra-subject variability, such as transit times, permeability variabilities, etc., on the absorption side, or clearance or PK parameters if you were going to plot AUC or Cmax values. So we have this view for the 20% free base. On average, the absorption is still like 95%. So 20% free base content is not projected to have a PK impact, or at least a meaningful PK impact. However, if we move to the 50% free base simulation, then you start seeing a more clear impact. You can see on the graph, the blue curves are the initial simulation of the 20% free base. The purple dots are the 50% free base, and clearly there is a differential behavior of these populations. A much larger portion of the dose is solubility limited, resulting in a sufficient shift on the mean of the profile. The mean here is about 85% absorption. So that would be enough for this formulation to be not bioequivalent to the original one. To take this one step further in terms of population simulations, all the previous simulations were conducted in a typical population that most companies will use for a relative bioavailability or bioequivalent studies. However, this percent of free base will have an even more pronounced effect in populations that are known to have a higher incidence of achlorohydria. So as a demonstration here, the initial 20% free base simulation was run also in a Japanese population, which is again the purple-pink dots on these plots. You can see that that population has a significantly higher portion of subjects with stomach pH above 5. For those subjects, the absorption is projected to be significantly lower compared to the rest of the population. As I mentioned, after these initial simulations, we did move the simulations back in GastroBlast software. We wanted to generate these parameter sensitivity analysis plots to visualize the effect better. And also we wanted to update the simulations to use some additional dissolution rate measurements that we had in the meantime generated for different formulations with different percent free base. The principles are very similar to what we discussed, so I'm not going to repeat all the simulations in detail again. The mean outcome of the simulation is also quite comparable between the two software. The sensitivity plots that are shown here clearly demonstrate how the percent of the free base content becomes an issue as you increase the stomach pH that is assumed for the model. Similar to the previous simulations, GastroBlast also at this stage doesn't allow for two API phases to be simultaneously present. However, since we know that the HCl salt fully solubilizes in the stomach, regardless of stomach pH, we can trick the simulation by using the mixed dose setting, where the portion of the HCl salt is dosed at time 0 as a non-precipitating solution, while simultaneously the rest of the API is dosed as a tablet or suspension of the free base. This approximation works pretty well here, again, because of the known behavior of the HCl salt. If we only had partial dissolution of the HCl salt, or the solubility in the stomach wasn't as high, this simulation would be much harder to undertake. So as lessons learned, while average simulations for the most part are sufficient for the most of the formulation decisions, population simulations and virtual trials are useful tools to understand the absorption variability and the potential interplay between formulation properties and physiological variabilities. As I mentioned, the software cannot simulate the multiple API forms. It doesn't have to be salt versus free base. The same question applies to potentially amorphous versus crystalline, for example. However, these initial models, even with the assumptions, were very helpful for us to understand what we're looking at and to potentially generate experimental data to drive further decisions. I will also admit that even if the software had the capabilities to simulate multiple API forms, understanding in vitro precipitation to in vivo precipitation connections, disproportionation, etc., is very difficult. So from experimental standpoint, to begin with. Again, I think despite these assumptions, these models are quite useful to inform either experimentation or subsequent clinical studies. So I'm going to close this presentation with case study 4, which is around developing an IV-IVC via absorption modeling for an immediate release product. The compound is a BCS class 2 compound with quite low solubility. It's 6 microns per ml. It's pH independent. The dissolution is sensitive to API particle size. Shown on the left, these are the dissolution curves for different API lots ranging from micronized to 1 ml material. In addition, we had a relative bioavailability study with some of these formulations. So our goal was to develop an IV-IVC using absorption modeling. We're going to use standard parameter input, which means basically dissolution rate measurements, solubility, permeability, particle size, and known IVPK parameters. At that stage of the program development, the IVPK were available to us. The model was built starting with the slowest formulation that had the largest dissolution or absorption limitation. That allowed us to estimate the in vivo solubility and the permeability numbers. During the model development, we did have to optimize some of these parameters to fit the observed data. The in vivo solubility estimate ended up being about 3-fold higher than our measurement in FASIF. It's probably within the potential viability of projections, although it's on the higher side of what we would like to see, especially compared to FASIF. The permeability prediction in vivo was pretty well aligned with our projections from KECO2 data. Since for an IVIVC, following current guidelines, one wants to be building the model starting on an individual basis. Some parameters, such as gastric emptying time, and some of the metabolic parameters, such as Km and Vmax, were estimated and assigned on an individual basis. These individual fits are very useful to understand the potential challenges of the models. Sometimes they are more telling compared to the average simulations. For example, I'm showing here on the left the same subject dosed and simulated with two different API lots. To be able to fit the data, we had to fit drastically different gastric emptying times for these subjects. You can see subject A, batch A, we used a stomach transit time of 0.8 hours. However, assuming particle size, solubility, and other things are the same, we can only fit the batch B data using a stomach transit time of 3.5 hours. This is probably outside typical justifiable ranges in terms of variability. Many other subjects were much closer. I'm showing the extremes here just to drive the point of this simulation. However, we know that transit time and dissolution rate will have a confounding effect on the absorption and the input rate projections from the model. So if we are fitting some of these parameters, our obvious concern will be, are we masking the actual dissolution rate impact? But for now, let's accept this model and this fits as correct, and let's see how the absorption looks. The outcome of the mechanistic deconvolution was quite telling in this case on the difference between the absorption time course in vivo and the dissolution in vitro. The lines demonstrate the estimates of in vivo dissolution versus time compared to the circles, which are the in vitro measurements. It's clear that in vitro dissolution for almost all the lots is complete roughly within 45 minutes, at most 1 hour, while in vivo dissolution takes much longer, with the exception of the smallest API. So obviously trying to do an IVIVC with this is not ideal. You get this hockey stick correlation if you plot fraction in vivo dissolution versus fraction the in vitro dissolution. There are mathematical ways to go about it, like time scaling, time shifting, and other manipulations. However, it's not an ideal scenario. So what we wanted to do is we went back and we developed a new dissolution method to reflect the in vivo dissolution process. So we switched from the standard QC method that employed some level of surfactant to another method that was more biorelevant and had the correct time frames. Just to avoid any confusion, by using the term biorelevant, I'm not saying this was passive. This was still not using passive or this type of biorelevant media. It was an aqueous-based system, however. The deconvolution that I showed you in the previous plot really helped drive this dissolution method development, as we knew what time frames to target. And also, it should be noted that on a relative dissolution basis, these methods give the same outcome from the different APIs. So we did not affect the discrimination or the rank ordering of the different formulations. So now if we plot this new dissolution method to these in vivo simulation curves, we are in at least similar time frames. They are not superimposable, but it's a much better place to be compared to the previous situation. So trying to establish the IVIVC with a model again, we're able to get a model that reasonably described most of the data. We still had to use some time scaling and time shifting in the model, and I will still admit it was challenging to get all the PK curves in the same line. However, if we only focused our model within batches that we were planning to move forward in the future, we got decent internal prediction errors shown on the right. I will admit that the external prediction errors weren't as good at that stage. However, the performance of this model that was based on the PBPK absorption model was not too dissimilar to a traditional IVIVC model we had developed in parallel. I'm not going to discuss the traditional IVIVC model due to the time limitations. So we had what appeared to be a reasonable model with potentially acceptable prediction errors, at least internally. However, we wanted to challenge the model and understand how unique these solutions were. So during this challenging phase, I've included here some plots of two individuals modeled with different combinations of solubility or gastric emptying times, where we went from the optimized model to using the primary data to fitting only one of them, and so on. You can see that the simulators are pretty sensitive to these parameters. So it becomes evident that the dissolution rate that we can assign as a unique parameter to the model may not be a unique solution. So the question is, how do we account for this variability? This variability is inherent to the in vivo data. This is even more difficult if one starts thinking about external validations. If we were to use this model for an external validation, and the gastric emptying time was a parameter in the model, how am I going to pick up what the gastric emptying time for an unknown population? So at the end, that does bring up the question, is the model sufficient for an IVIVC argument or not? Lessons learned. We only have another 3 or 4 slides before we open up this for questions. In our experience, full bottom-up absorption models are not easy to put together. There is always some need for fitting of the data, and part of this is due to the inherent error from projection from in vitro data to in vivo data. Even things we take for granted like permeability or solubility projection from in vitro to in vivo, if you look at these correlations, there is always a couple of fold error in some of them. So the question is, if we are to develop these models for IVIVCs, how do we defend these parameters? And what is considered an acceptable value? I will also say that standard IVIVC approaches that are currently acceptable do not fully account for all these aspects of variability. So the variability question is not only unique to the absorption-based IVIVCs, but it does become more obvious probably because of the way the models are set up. For this specific project, I will say that the absorption model gave us, at least internally, assurance that our correlations were meaningful and that our traditional IVIVC was also meaningful. In the end, we did opt to use the traditional IVIVC for any of our arguments. But I think it does remain a question whether a fully validated IVIVC, at least based on the current guidelines, can be built using absorption modeling. I'm going to close this webinar presentation today by recapping some of the challenges that were discussed on the application of the current absorption models. At the same time, I see these as opportunities for further research and further discussion. And part of the intent of this GastroPlus user group is really to push towards solutions in these areas of common interest. I really hope we will continue discussion on these topics either via the user group or on our LinkedIn website. Historically, these models have been applied to conventional formulations, crystalline dosage forms. Even for those, we need to start accounting for more complex dissolution processes, past the API PhD diffusion-based dissolution model. However, the major gap really comes when we start tackling the behavior of more complex dosage forms. Soil dispersions, nanosuspensions, lipid formulations that are commonly used for BCS class 2 and 4 compounds. Having multiple API phases present, particle size generation upon dissolution, crystallization and precipitation are questions that are not easily answered either experimentally or easy to put in the models. At the same time, there is also a higher level of challenges I'm going to touch upon related to the application of these models. In early development, the main constraint is data availability. One really needs to balance the data and the assumptions. The willingness to do that is really modeler specific and organization specific. However, in late-stage development, and especially for regulatory applications, additional points need to be considered. First of all, model validation and the lack of a broad agreement of what model validation may look like. How does one justify parameters versus experimental data? There is a traditional IVIVC guidance where we do internal and external validation. Should this be an expectation for PBPK absorption models? When does the model become too complex to justify? We always want to use the simplest model possible. How does one define complex versus simple? The second one is translating this to a current quality control practice, which is what will actually impact the product at the end. Having the models is great. We all like nice models, especially we're the modelers. However, if we cannot translate it to practice, there is not much use of the model. So when we're talking about quality by design, a lot of the times the endpoint is dissolution method. Translating the model outcome to a dissolution method for a routinely manufacturing process in a quality control setting is not really straightforward, especially for insoluble compounds. And finally, we need to keep in mind the diversity of regulatory agencies' opinions on models. What may be acceptable or expected from one agency may not be the same with another one. However, despite these challenges, I continue to believe that absorption modeling has a significant role to play in quality by design. Even at the end, the model is not positioned to make a direct impact on a specification or in the filing of documents. From experience working across several compounds, I can guarantee that it will have helped promote collaboration and promote cross-functional understanding. The quality by design schematic I saw at the beginning highlights this collaboration and knowledge sharing aspects. Translating the dissolution curves that are shown on the left to the plasma profiles on the right is really a very powerful communication tool. Most of the people on the development teams, unless you are the person who generated the data or perhaps the formulator, won't really be too interested in dissolution curves. However, they will be very interested in plasma profiles. So even if the model is translating something that is obvious to the modeler, it facilitates acceptance of these concepts and across very diverse functional areas. So I think, in my experience, if you want to take something from this webinar as a closing point, is that this collaboration and this ability to translate the data to other functional areas through modeling is very important to achieving the quality by design goals. With that, I want to thank my colleagues and collaborators whose work was included in the slides. And with that, I'm going to turn it back to Charlene so she can facilitate the discussion. Thank you for your attention.
Thank you very much, Philippos, for the great presentation. At this point, we will open up for questions. I've seen that we already have questions coming in. As mentioned earlier, you have two options to enter the questions, either through the questions pane on your control panel or using the hand raising icons. So start with our first question. The question is from Mark. Do you always refine in vitro dissolution methods after getting phase 1 PK in human? In another word, do you always try to fit deconvoluted PK by the in vitro dissolution method?
No, we don't always try to do that. We use a range of methods ranging from biorelevant dissolution to more QC type of dissolutions. Since the biorelevant dissolution methods are already accounting for the physiological parameters, a lot of those measurements drive our formulation development early on and go into these models. If we're talking about the IVIVC specific part, if we get to the development of the IVIVC, then we do look whether there is a potential for refining the dissolution method.
Okay, thank you. The second question is about your case study number 2, related to case study number 2. Was it assessed if the 120 milligram of the presented BCS class 2 drug soluble in 250 milliliter at pH 1.2?
Yeah, it's freely soluble at pH 1.2. The solubilities are shown here. So the solubility below pH 2 is 25 mg per ml.
Okay, thank you. The next question, is the dissolution data at pH 1.2 clinically relevant for the drug that you examined or for all data? Is it premature to have this conclusion?
I'm not sure I understand the question about the all data part. So the claim here is specific to etoricoxib. I can guess, well, I mean, I have experience with other weak bases that pH 1.2 could be the most relevant condition, but there are also weak bases that pH 1.2 is not the most relevant condition because of precipitation. So I think it has to be judged on a case-by-case basis, if I understood the question correctly.
Okay, thank you. The next question, what type of formulation was used in case 1? Was it viscous?
No, it's a standard granulation with crystalline API. I don't recall the specific composition on top of my head right now.
Okay, thank you. The next question, can you, okay, the screen is moving. Can you please expand a little bit on what parameters were different in your new dissolution method that reflected closely on BA, on the bioavailability?
So I assume that refers to case study 4. So the difference was the quality control method, the initial quality control method was employing a specific level of surfactant. So that surfactant was minimized or removed for the final dissolution method.
Okay, thank you.
It was a common surfactant. It was a type of SLS, or I believe it was SLS.
Okay, the next question. In case study 1, in your first case study,
yep,
the delay in dissolution of formulated API is really a common rather than a question. In case study 1, the delay in dissolution of formulated API is disintegration related.
So as far as the micronized API goes, it's probably disintegration related. But I will say that in general, the point we were trying to make here was look at the dissolution of the formulated API, because I've seen products where actually smaller particle sizes result in slower dissolution. It really has to do with how the formulation is made. So Just what I'm recommending is not just use the API-based particle size model, but also look at the dissolution behavior.
Okay, thank you. The next question, have you seen the difference of huge dissolution rate between in vitro and in vivo due to large buffer effects in vitro, but not seen such large buffer effects in vivo along with a smaller volume of solvents?
That's a good question. I cannot say that I've seen. Well, I guess it depends how large differences are defined. I do believe that buffer has a significant role to play for salts, especially for salts of weak acids. And I think there's some very good publications out there, some of them from Gordon Amadeau's group from Michigan, and also a couple of them from AstraZeneca that study the effect of buffer capacity on salt behavior. But I don't have any specific examples that I can think of immediately that the disconnect was huge to impact a model right now. Perhaps because we didn't study a compound that would have this effect.
Okay, thank you. The next question. First, good presentation. Thank you, Phil. Your conclusion seems like based on all BCS2 compounds. How would you think if this applies to other BCS drugs?
Yeah, so I think BCS1 compounds, I don't think anyone has too much of a concern about them. I think everything applies. Unfortunately, most of us on the call don't have BCS class 1 compounds to develop, so I think BCS class 2 and BCS class 4 are what we are mostly dealing with. It has to be looked at on an individual basis. The BCS class 4 compounds are much more difficult to handle if you are really having a true permeability limitation, because I think those projections are even more difficult. But I do think it has to be looked at a compound basis, and given the data you have at hand and whether you can build sufficient knowledge to drive a model.
Okay, thank you. The next question, could you comment more on the particle size distribution effect on API dissolution in API and formulation? If they do not match, is this due to formulation manufacturing process, that is a hot mill or wet granulation, etc.?
Yeah, so in this case, these are simple granulations, so there is no hot melt, so we're not dissolving the API. I've seen data, similar effects also with granulations that do not involve dissolution of the API, or at least not complete dissolution of the API during the process. Some of the times, depending on the process, we've seen breaking of the API, and that's one possibility. So larger particles become smaller. Some of the times, I've seen that the API controls the density of the resulting granules, and the granule actually controls the dissolution. So if the density is high, you might have a slower dissolution just because of the granule properties. So it is related to the manufacturing process, but it has to be looked on the compound basis and exactly how you're manufacturing things.
Thank you. The next question is regarding case study number 3. Can you please repeat how did you manage to predict profiles of formulations with different percentage of base?
Okay. So for the Simpson simulation, let me go to that slide. What we did was a stepwise simulation. We simulated the free base part as the hydrochloride part as a non-precipitating solution. So if it was like 80%, let's say, HCl, 20% free base, we'll get 80% of the dose as a hydrochloride salt. Then we'll do another simulation of the 20% of the free base, a completely separate simulation, and then add those two together. There is an assumption happening here. For the gastropro simulation, because we can use the mixed dose model, and I might not be pronouncing the model correctly. Vieira or some of the simulation class can say the exact name of that function.
I think you got it. I think you got it correctly. It's the mixed multiple doses. Yeah.
Okay. Thanks, Vera. We put at time 0, 80% of the dose as a non-precipitating solution. So we put the precipitation time in the model to a very large value. And the time 0 again, another 20% of the dose as a free base. So that part of the API as a suspension. So that part will dissolve based on particle size and solubility numbers.
Okay, thank you. The next question is, after establishing and validating a good IV-IVR based on the solution and the clinical data, how to decide about the relevance of in vitro method in predicting a new batch if the F2 turns out to be around 50?
Yeah, yeah, that's a very common question. So I think it depends on, I guess, what type of IV-IVR you have established before. If it's a computational type of IV-IVR that will be closer to the IV-IVC, obviously you can just use that model to drive a prediction. If it's more of a semi-quantitative or qualitative rank order, I think it really depends on what differences you've seen in your clinical data compared to the dissolution differences. I know there are some very nice presentations given in the past by FDA biopharm colleagues around using clinical data to understand clinically relevant specifications and impact of bioavailability. There is also the safe space concept from AstraZeneca that has been published before. It has to be looked on the compound and the dissolution data and the clinical data you are looking at. I will say that the minimum requirement to start looking at this will be that the process changes you are making for this new profile has been covered in your previous clinical studies. I hope this answers the question. If there's a follow-up, someone can follow up either now or via email later.
Thank you. The next question is for GET or STT. I presume that's gastric empty time or stomach cleansing time. It's about 1 hour generally acceptable. I assume it is a fasted condition. If so, it seems so long.
Yeah, so that's a good question. So the default, well, I guess the default, well, not the default. So the measured gastric emptying times for solutions or reasonably sized tablets range somewhere between half-lives of 15 minutes and half an hour. So if you look at the entire variability, I think 1 hour is still within the potential variability of reported data. The mean should probably be earlier than that, though. So I agree with the comment, but I do think it's within potential variability.
Okay, thank you. I saw a the someone raised their hand, and this is the previous person who asked these questions. Let me see if I can unmute. Hi, Li Ping. Oh, I'm sorry. Hi, Li, you want to go ahead, ask your follow-up questions? Okay, never mind. I didn't hear anything. Let's move on to the.
Yeah, and again, I mean, as you said, if people want to discuss anything further, please free to post a question on the LinkedIn website, and we'll be more than happy to answer there.
Oh, okay. So I saw a message. Hi, Lee said it's already addressed already. So that's good. The next question is, can you comment on use or success of using GastroPlus Z factor?
Yeah, so the Z factor model is basically a manipulation of the entire baseline Johnson model by removing the particle size factors. In my experience, I prefer using the Hinz-Johnson model by including some fudge factors if needed, like for example on the diffusion coefficient, or by generating hypothetical particle sizes, because it gives more flexibility to capture a wider range of dissolution profile shapes. The Z-factor model does constrain you to something that will look closer to a first-order dissolution profile, just because of the way the math works. I don't think there's anything wrong with the model. As long as you can describe your data, you should be able to use the Z-factor model.
Okay, thank you. The next question, in case study 1, how did you incorporate your dissolution profiles, parentheses API powder or formulation into GastroPlus, or whether you simply manipulate the D effect with the built-in DISO model?
Yeah, so for the API, so we simply manipulate the D effective on the DISO model. So what we did was we changed the physiology in GastroPlus to simulate dissolution in a beaker. So, barring hydrodynamic issues. So, based on that, we calculated what the D effective should be for it to match our profiles, and we use that D effective values for the simulations. I guess if people are using the DDD Plus software, they can do that part of the simulation, find the DDD Plus software, and then move those parameters in GastroPlus. We were just doing it directly in GastroPlus.
Okay, thank you. The next question is, can you please inform if any literature method has used the inline changing over of dissolution fluid in a single experiment, say SGF changed to SIF?
So the question certainly says inline changing?
Yes. Can you please inform if any literature method has used the inline changing over of dissolution fluid in a single experiment? For example, from SGF change it to the SIF.
Well, I guess if the definition of inline will be that, you have the single source that the dissolution fluid will go through exactly the same beakers. I believe there is some publications using the USP4 methods, but from what I've seen, most people typically will, who are more after biorelevant conditions, move towards, these days, towards transfer type of methods, where you have dissolution in the SGF happening in one compartment, and then you transfer that to a compartment where the intestinal fluid exists. I think that's where I see most people going to, but I believe there are USP4 protocols that involve inline changing of the media.
All right, thank you. The next question is, do you have any experience to use this software to extrapolate different species, that is from preclinical models to human models?
Yeah, we do routinely run some simulations between dogs and so for formulation development we mostly use dogs, so I'll refer mostly to dogs. We do simulations both in dogs and in humans. I won't say that they always agree, and part of this has to do with whether you have dog-specific parameters to go into your model. You need to think about specific solubilities, differences between species, and potential permeability differences, depending on what you think your species is doing relative to humans. But yeah, I mean, I've seen plenty of models where there's good agreement in the extrapolation of the formulation behavior between dogs and humans, and I've also seen a fair set of models where dogs and humans behave differently.
Okay, I think Vera knows the most about this software. Do you have any comments on this one?
As for the extrapolation, there have been number of articles actually coming out on extrapolation across the species. In fact, one of the most recent ones was from Philip Ozon showing the extrapolation, and there were some other references as well. So it may be a good resource to look at our website, where we list all of the latest references showing how the preclinical species have been used to extrapolate to human. I know that some of our other customers, I'm not sure if any of them are online right now, can see the name. Some of it may not be published. We have got some feedback, especially on the Z-factor model, that one of the uses of that one, or one of the reliability of that is that it works well, but warning was it should be really validated or verified that for given formulation it works. And using preclinical species really seem to give these customers good way of validating that the model really works and translates well.
Okay, thank you, Vera. The next question is, have you seen any drugs dissolved faster in vivo than when in vitro, especially in for weak-based drugs or products with salt forms?
If I've seen faster in vivo compared to in vitro?
Yes.
Well, I guess it depends on what dissolution method you do the comparison. If you compare against a QC type of method, I have a very hard time thinking of a case where in vivo dissolution will be faster than the QC method, just because you have to do with gastric emptying time in vivo. If you refer relative to biorelevant methods, yes. I mean, I think there are cases, and I've seen cases where in vivo dissolution appears to be happening faster compared to in vitro, and some of this might be related to buffer effects, etc., as discussed in the previous question.
Okay, thank you. The next question is how would you define the solution correction factor?
Yeah, so for the case study 1, the dissolution correction factor was only, a multiplication factor on the diffusion coefficient to allow to fit the data. So for example, if the initial diffusion coefficient, which was molecular weight based, was about 6, we multiply by, I don't recall exactly, like 0.2 or something to make it fit the data better.
Okay, thank you. And that appears to be our last question. So let's see.
Actually, it seems like Mary Pan has her hand raised.
Okay. All right. Let's unmute. Mary Pan. So anybody online, you can go ahead, ask your questions.
Mary, you still have question?
Can you speak a little louder?
Okay.
Okay,
I think the question has already been answered. So.
Okay, very good, very good. There's another person, Amusa. You have your hand.
Thank you.
Okay, go ahead.
My question is somehow general to bioavailability assessment. I don't know if the presenter can give brief explanation. I was simulating an in vivo data, and I found that the model describing kinetic parameters for capsule was specifying different model than the one for tablet. Say for instance, tablet was saying one compartment model is the best fit by SIC, skills, and the other such parameter. And the capsule was saying two compartment model was the best for the same drug. Is that possible? Has anybody observed such?
Yeah, so okay, without knowing the details, it's difficult to comment. But so generally speaking, the first thing I would look at that case was whether you have some type of delayed dissolution of one of the, I guess with the capsule, which is your slowest one,
Okay,
and that
yes.
results in you having a pseudo second compartment that mostly reflects absorption rather than actual disposition.
Okay.
I mean, I guess under extreme cases, if you have dissolution differences, and you have saturable distributions, you could potentially alter systemic distribution, but I would think those cases will be very few, and it will be quite unusual to have such a drastic effect. If you are seeing different profiles from different fits from the different formulations, one way to understand what's happening might be to try to do some deconvolution type of analysis and see, using the faster formulation as a reference, and see what is it telling you for the slower formulation.
Okay. 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 Thank you.
All right, thank you. I saw another question from Jasmina.
Hello.
Go ahead. We cannot hear you, Jasmina. Can you speak a little louder?
I'm not sure that I have technical possibility. to talk, but I already asked 2 questions, so both my questions have been answered already.
Oh, good, good. That's great. All right.
Thank you.
All right. If there's no more questions, I also see that our time is over. I think we reached the end of the time for the webinar also. In case you have additional questions,